I'm trying to go paperless, so I had to bring these lengthy introductions to my co-panelist here on my iPad. So thank you for sticking with us. It's been a really amazing day. It's been like a whirlwind of really good science, really good thinking. And we're going to close today with, I think, really a very practical discussion, which is how do you get the science into the clinic and ultimately complete a clinical trial that will lead to some type of regulatory approval or access approval. I mean, the numbers that I have been given in my new role at Lilly is that over the next 5 years, in order to complete the greater than 1,000 trials that are ongoing in IO, we would have to identify close to 700,000 patients to enroll in studies. So even you think about it, I'm not super good at math, but 1,000 studies, how do you get the right patient on the right trial? And so that's really the purpose of the next 35 minutes. What I'm going to do is introduce my panelists, and we're going to have a discussion. And then I just want to preempt my introductions with, I do want to have an opportunity for people to ask questions. So I'm going to kind of stop the panel and allow for about 10 minutes for people to ask questions to our esteemed panelists. So I want to introduce Dr. George Sledge. Most of you are very familiar with him. He's a professor, chief of medical oncology at Stanford. He's a world-renowned breast cancer leader. He has served on ODAC. He is a member of the External Advisory Committee for the Cancer Genome Atlas. And although I don't see it on this biography, you were ASCO president, which was left out of the biography on the website. So that probably is one of his largest accomplishments. It does list you as chair of ASCO's education committee, however, 1 year. So thank you for joining us. And then Theresa LaVallee, who I met while we were working on a HIF-1 inhibitor like 25 years ago. So that tells you how long she's been kind of slogging along on innovative approaches to treating cancer. She's currently vice president of the translational medicine component of the Parker Institute for Cancer Research. She previously served as a vice president at Celldex, Coltan, MedImmune, and Entremed. So really a lifetime of commitment to bringing drugs to patients. And I think we'll start there, and I'll start in the middle. So if that's okay. I'm not going to stand up there. All right. All right, so we have kind of the three pillars of the ways that drugs are accelerated in bringing new medications to patients. So I'm going to represent the sponsored or the pharma perspective, my long 6 months in the industry. George, you're going to talk about kind of an academic perspective of how do we bring new IO agents in particular into the clinic and this problem of clinical trials. And Theresa, you're going to talk about really the innovative model that Parker's using, which is identifying, well, you can talk about it. So we didn't draw straws, but maybe we'll go in order of the way we're sitting, and we'll start with George and kind of really touching upon, pure academic clinical trials like the one we just heard of. What are the really positive aspects of that, and what are the limitations? And I mean, there's some obvious ones, but maybe you could start there.
Thank you. And Kim, always a delight to be on stage with you.
Thanks.
So, I think a kind of a good starting question for all three of us is, what are you guys good for? Okay. Because I think whether or not you're talking about pharma, either big or small, whether or not you're talking about foundations, whether or not you're talking about academia, they all have strengths and they all have weaknesses. The strengths of academia are pretty straightforward. First, you have lots of assistant professors. This turns out to be incredibly important because these are the hungriest people on the planet, the ones with the most brains and the ones who are most up to date on the most recent technology, and therefore the ones who are most likely to create something new. These operate within an environment where drug discovery is not the endpoint of what they're doing, but discovery is. And institutions such as Stanford are great at discovering basic science. that subsequently becomes applicable to the clinic. What we're not so good at, we're not good at a lot of the things that pharma does well. We're not good at all of the I&D enabling stuff, like toxicology, for instance. We're not, some places are good at pharmacokinetics and pharmacodynamics, but many are not. But we are really good, we're really good at finding what represents appropriate populations to treat with particular novel therapeutics. And this, again, comes largely from the discovery that is done within institutions. If you ask, is it possible for academia to bring a drug to the clinic that started as a research enterprise in a laboratory within an institution? The answer is yes, but it doesn't happen very often. An example from Stanford, for instance, in the IO space is the CD47 study, where Irv Weissman and Ravi Majeti did basic research demonstrating that CD47, the so-called don't eat me signal, that cancers co-opt to avoid being eaten by macrophages. Laboratory research done at Stanford showed that if you masked CD47 with an antibody, that in turn allowed the macrophages to wake up and eat the cancer cell. Through a gargantuan effort by Irv Weissman, Ravi Majeti, and many others at Stanford, we were able to carry this all the way through into phase 1 and a phase 1B expansion, which led recently at the most recent ASCO meetings to a wonderful presentation by Stanford's lymphoma director, Ranjan Advani, to show a very high clinical response rate in a population of refractory patients. It's a great example, but it's also an exceptionally rare one. And it's rare because being able to put all those components together, I would say, is not the forte of academia. The other thing we're pretty good at is doing phase 3 trials. And we do those through cooperative oncology groups. Throughout most of the history of modern cancer medicine, most of the phase 3 trials that led to an approval by the FDA were done through the cooperative oncology groups. This has fallen apart in recent years, in part due to the fact that it takes fairly long for a cooperative group to do this compared to a pharmaceutical company or a CRO. But also, I think, the increasing desire of a pharma to control its own fate rather than turning it over to what is, in essence, an academic government partnership.
So good at a lot of things.
But not good at everything.
Yeah, not good at everything. So I'll just give two minutes on the pharma perspective, which, you know, when I was thinking through today's panel and having come from the academic sphere now 9 months ago, there are some things that pharma actually does very well, and I'm speaking on large pharma, but you could also apply some of this to appropriately funded small biotech. The one thing that large pharma and small biotech, I think, share is that you have to have a laser focus on what you're trying to accomplish. And I think in some ways that it can really serve clinical trial design very well, this principle of proof of concept and how do you achieve it early on, I think is applicable to both small biotech and large pharma, which is really what do you absolutely need to see in a clinical trial to deem that drug worthy of additional resources and ethically not putting patients on drugs that don't have some biologic or clinical activity. So I think the laser focus, not that academicians or institutes don't have a laser focus, but I think the reality is that you're held accountable to the stakeholders, whether it's large pharma or small biotech. Resources, I can tell you from a year ago to now, the amount with that laser focus is as long as it's achieving a goal, some of the resources for good and for bad that we were able to apply to clinical trials is really pretty amazing, and I think most people recognize that. What we're not so good at is how do we utilize those resources to inform the next trial. So assets are typically developed, at least in my experience thus far, in silos. So things that one team will have learned with one IO agent might not even actually be transferred to the other IO agent. Large platforms of data across the history of Lilly Oncology or any large pharma company. I think we see some pharma companies doing extremely well. We just saw a paper from the Merck group showing their long history of, basically compiling the data. So I think sometimes there's a large amount of resources, but they aren't necessarily used in a very efficient way. There's not a learning environment. And I think many people are trying to change that both in small biotech and large biotech. The third thing that I think is really good is, and you touched on it, is really this pre-IND and IND capabilities that really exist within either small biotech utilizing a CRO, a clinical research organization, to do their preclinical talks, or actually within a large pharma. And having just returned from JP Morgan like 2 weeks ago and seeing 86 companies that all are plugging different types of cellular therapy and non-cellular IO therapy, it's pretty amazing how that can actually be a kill signal when you can't produce in a large scale, not just even T-cell therapy, but the actual product. And so the capabilities and the scientists that go into that, I think I was definitely very naive as to what it actually takes to get a drug into an IND in a phase 1 study. I was always just kind of handed it on a silver platter, which in retrospect seems really nice, and not really appreciating the resources that went into it. So I think resources certainly help. I am quick to say, especially recently, that you can throw a lot of money at the problem and you can throw a lot of science at the problem, and it's still not fixing the problem. And so I've seen both really good science from the academic and in the industry, but also a lot of resources being spent. And I think it's really going to be that sweet spot emerging. And I think we heard about it from Carl June and other people today, where magic really happens when you have the resources to do what you want to do, but the science ultimately has to drive the process forward in a clinical trial. So, and I'll turn over to Theresa, because I think the Parker Institute, to me, represents really one of the ideal ways that we're going to move the needle on the IO field. So maybe you could say what you think you guys are doing well and other institutes are doing really well, and where there's some areas for improvement.
Sure, thanks, Kim. And the Parker Institute for Cancer Immunotherapy, I think, is very unique in that it tries to accelerate learnings in immunotherapy with really the sole mission to cure cancer with immunotherapy. And what it's tried to do is really answer gaps. What we aren't is we're not pharma. We're not an IIT, we're not a sponsor. We're kind of a blended model that's really trying to use science to learn, to inform, and within patients, bring medicines to patients. And so the overall concept is through collaboration, bold research, so nothing redundant. So the thousands of therapies that are on clinicaltrials.gov, and CRI has done a really nice job of really doing landscape analysis, as if it's PD-1 and LAG-3. That's not necessarily interesting, that there's a need for us to do something different. If there's a unique biomarker or something, we would do it. And so it's doing outside of pharma, outside of academics, of really bringing people together. And I think that Carl June and your questions with him this morning and many of the talks today really set up nicely, where he mentioned bringing in multiple modalities. So these ideas of how do you bring together a CAR T, a microbiome, a checkpoint inhibitor, et cetera, across multiple companies if they come from different places. And that's really what Parker's tried to serve, is to look at the science. If there's a really interesting scientific hypothesis that hasn't been done in tumor types that are cold or underserved, So Bob Vanderheit's work with CD40 really looking at the sequencing with checkpoint inhibitors, showing that in a lot of preclinical studies and some early clinical studies that he could see responses with CD40 antibodies in pancreatic cancer. And so Parker brought together BMS and a small biotech, Apexagen, to do a clinical study, a multi-center clinical study with our 7 institutes to give industry quality data. So we hold the IND. We don't do, for those types of studies, they're not IND enabling. Having a bunch of us that have pharma experience, we can do enough to give a rigorous package without taking a lot of time to get to an answer. But the overall concept is, as we run the clinical study with source data verified data, that if we see a signal, pharma can take it and hopefully apply for breakthrough therapy designation, design a pivotal study. So it's industry quality IIT type studies that are taking innovative approaches, bringing multiple companies together, looking at sequencing or different aspects. And importantly, within those studies, we're doing deep immune profiling. So we really interrogate the immune profile of each patient. And so another thing that Carl brought up this morning was the need for tissue. We've seen many examples and talks today about tissues. In pharma, there's always an interest in getting biopsies, but then when you look at global studies, when you look at the costs, when you look at enrollment curves, they're inconsistent with fast-to-market strategies if you have to require baseline and on-treatment biopsies. We have the luxury of really wanting to learn why it worked, and also importantly, what I think after spending 2 decades in pharma, the one thing that isn't done well there is why didn't it work? So we look at a variety of different approaches to really bring in data sets, and by having standardized collection across all of our studies, as well as a clinical database and a biomarker database that are integrated with harmonization of collections and time points, that we hope to do the multi-omic analysis that the Merck team just published on, as well as working with our academic centers who are providing data sets and our pharma partners who are providing data sets, and asking, can we bring in large multi-parameter data sets to learn more about why things have worked and not worked? So one of the things that we've been challenged with by a number of pharma that are interested, as well as many of the Parker academic folks that we work with, is a PD-1 resistance study with novel combinations. And we specifically have not done that because we don't know what we can do differently. So there's hundreds of thousands of studies ongoing looking at combinations and ways to address it. And so the way that we've approached it is to kind of say, what can we learn to do something that isn't being done? So can we work with the SITC Timios team that are characterizing super responders and non-responders with a laser-focused genomic approach? Can we bring pharma together and ask, can we collect data sets to understand, to then set up patient segments? So if we're hearing TCR clonality is predictive for response. I've seen lots of talks where people have talked about CD8 being predictive. This is another example of where everybody will tell you that's how you identify a hot tumor, yet no one's ever done the study. So we have a clinical study open now where we're stratifying, making treatment-based decisions based on CD8. So if you're CD8 high, you get PD-1. If you're CD8 low, you get PD-1 plus CTLA-4. And the idea is if it works, let's use that as a criteria for monotherapy, and then look at how we can do combinations and mechanisms of resistance to inform combinations to do other treatments. So it's a science-driven approach, which is, as I've worked with these guys for 20 years, I remember George and I had a debate once about whether to use biomarkers or not, where you took the contrary, despite doing mamma print and all of that. But I think biomarkers are the answer that will get us there, but it's a complex algorithm. It's not a single biomarker for a single treatment. So.
Yeah, and I look externally to Parker Institute and think, well, none of what we've talked about today are perfect solutions, but I think we would all agree, moving into the new era, that we've got to do things differently. And I think both academics, pharma, and even the institutes that you see emerging, the fact that you're actively saying we're not going to just be doing the same clinical experiment that 1,000 other groups, pharma, academics, are doing, I think that's a pretty bold statement, because I think I speak for myself and all of us that we've got to come up with different innovative models to speed this process up. And I think we heard it at the beginning of the day with Dr. June talking about, this is just the end of the beginning. And so I'm really excited to think about the other models that each of these entities are coming up with. I'm going to put George on the spot. We'll go back to this end.
Sure.
And I ask each of y'all to think about what is is there a trial that's perfect? Have you actually seen it and your jaw just kind of dropped and they said, that's what we should be doing? So I'll put you on the spot.
Well, I've only been doing this for 35 years,
okay.
so.
Well, let's talk about recent.
So that's probably not long enough to see a perfect trial.
Yeah.
I've never seen a perfect trial.
Okay.
I've seen lucky trials.
Yeah.
I've seen trials where the investigator tried a particular drug, a particular combination, it worked famously. But I don't ever know that trial was designed to hit a home run.
Yeah.
They rarely are. You were saying we need to think differently. What appalls me about what's going on right now, and by the way, I admire the Parker Institute for trying to break out of this. What appalls me is that we're doing every single mistake we ever did in the past all over again, but on a larger scale. You said 1,000 trials. I've heard 2,000 trials. But whether it's 1,000 or 2,000, do you really think that there are 2,000 or 1,000 good ideas in oncology right now for combinations? Instead, what we're seeing is we got a couple toys that we can play with in the sandbox, so we're going to do it. That's pretty much what we've done so far. And so the huge number of combinations that we're seeing are, by and large, are underpowered. By and large, do not have appropriate biomarker analysis attached to them. I'm in favor of biomarkers when they're done right. The problem is that they're usually not done right. They're usually done on insufficient samples, and the statistics kill you over and over again. And I see this happening again and again in our field, and I think it's a major disappointment. I would imagine in part, again, by the assistant professor's desire for CV polymerase, in part by pharma's desire to make the next big score. But regardless, I think we're not doing it well. And not only are we not doing perfect trials, we're doing an immense number of imperfect trials.
Oh, wow, that's a bummer. Okay, I thought you were going to come up with something really positive. Do you want to take a crack at that and see? I mean, has it been a moment in the past couple of years where you saw a trial and you just said, they got it right? Maybe
Well,
not the outcome, but just the approach, maybe.
And I think George, as usual, has hit it in that the pain points, and I think the challenge to people is to listen to what's not working, and so, and I think that we don't need to look for perfect. I think we need to look for something that we learn from, and I think that's fundamentally, for me, translational, having done it when it was experimental medicine, learning and having a clinical hypothesis is the most important thing. So when you go in, and it's really interesting because we've set up platform studies. So we're opening one in castrate-resistant prostate cancer a cold tumor that hasn't been highly responsive to immunotherapy. And the idea is, with this very wonderful and esteemed group of investigators, it's BYOC, bring your own combination. So what's the most innovative science you've seen in your lab, in trials, heard at conferences, and you would bring together multiple different treatments in combination. We'll bring the drug companies together and run the clinical study, and hopefully across the arms, if the biomarkers are standardized and we're looking, we'll be able to learn something to adaptively inform when we expand. Maybe it's add another drug, maybe it's change the sequence. It's those types of approaches that they're small and underpowered, but together they're designed to really learn and take into a cold tumor and try to figure out how to make it, what would make it immunologically responsive. And I think it's those ideas of asking people, though, when they come forward to say, oh, you should put Ipi-Nivo together. Well, why? What's the hypothesis? And I think if we demanded everyone not to tell us what the combination is, but what's the hypothesis? So if your hypothesis is you need to drive T cells in and CTLA-4 will do that, then did you study that? And really looking, I mean, so we are trying to do a study as well, and we're using microbiome treatment based on the work that Jen Wargo published last January in Science of looking at the profile of the microbiome in complete responders versus non-responders was different, and if she collected the poop from the cancer patients and put it in mice, she could change a non-responsive animal to a responsive animal. Well, we're translating that to the clinic in a year to look at it, but to think that we're going to do the first microbiome study and knock it out of the park isn't, it would be wonderful. I'm sure that everybody would love to see that, but what it's really designed to do is to say, can we get engraftment? Can we change the microbiome of a cancer patient by giving them FMT? Can we drive T cells into the tumor? Do we see good safety? Do we get hints that it will tell us what type of patients? Not every patient has a bad microbiome, so who should we be treating? And so the trial is designed not with pharma metrics and get to this POM, POP, POC. It's really is this treatment modality an approach that's going to help us overcome resistance, and what can we learn from those cancer patients to really inform how to do it in a more directed way? And I think, you know, that's been my passion my whole career, is use the science, and we can learn better from the science. And I think when we do that, we see good outcomes. So some of the lucky ones are, I think, sometimes very directed, science-driven therapies and trials.
Yeah. So what I'm hearing you both say is that the perfect trial probably doesn't exist, and we shouldn't let perfection hinder our desire to make certain it's good and it has a good hypothesis. And although I don't think we're supposed to call it poop, just so you know. It's highly selective fecal transfer or whatever y'all are doing. So I was told that you can't call it poop, apparently. You know, I'll take a little bit more of an optimistic, because I perceive that we've put a lot of resources in the past 20 years into drug development that hasn't necessarily amounted to a lot of breakthroughs. And, you know, one of the things that, you know, drove my own personal decision to enter pharma was if we continue at the pace that we were continuing, there would be maybe 2 more breakthroughs in breast cancer before I retired. I'm not planning on retiring until I'm 70, so that you can figure out how old I am. But
29.
yeah, that's right. So we got a long time to go. No, so, but I mean, I think the reality, the thing that I see is we have so many more tools in our toolbox, and with not a lot more resources, just smart use of the tools, in particular the biomarkers and the predictive markers. I think we're going to speed this up very quickly. And I think the companies that, in particular, and embrace this idea that we have to have a community, and let's not forget about patients. We haven't talked a lot about, we probably, in hindsight, should have had a patient sitting up here saying, what are the patients doing that help this effort, and how can they do better, and what have we not done, to foster that. But I do think that the toolbox is pretty full right now, and asking the right question with the right tools. When I look back 25 years ago, I remember doing a trial with you where we were doing sequential biopsies with single agent paclitaxel. And I mean, we were really so bad, we were having to freeze the tissue and, section it, and the tissue would be really kind of crappy, and the antibodies were really crappy, but we were trying at least. And I think the reality is the tools that we've heard today really will foster a better interaction. So I want to make certain that we have time for some questions. I just have one more question for the panel, which is, with this number of patients required to complete some of these trials, and you only get one answer, so you got to think quickly, but what do you perceive as the one major barrier? Okay, you can have two. I'm going to give you two. But, maybe I'll start with you, like, just from your perspective at Parker Institute, besides science and besides, money, what are the real resource, the real barriers that y'all are thinking carefully about every day to try to really foster accrual to the trials and collaboration? How do you foster collaboration or break down barriers, and maybe you can't answer it in two, that's probably a bit unfair, but what are the things that y'all are thinking about every day?
Well, I think in terms of getting the people, getting the accrual and getting good metrics. I mean, Parker's only 3 years old, so the N is still small, but what's been a joy to me is this idea of collaboration. So it's the investigators' idea, and they all work together as a unit. So our enrollment actually has gone much faster. So, usually the enrollment curve projected and then actual, whereas ours is actually going faster because I think that everybody's bought into the study. It's not a pharma sponsored, well, here, you do this study. It's their study that they work, and pharma's at the table. So it's like, when I try to explain to people how we work, it's a cross-section of the pharma, the academics, and Parker proper, Parker Central, where we are holding the study and the data and the IND and really trying to execute, operationalize the study. And I think it's us working together that's really galvanized that look and that interest in trying. And I mean, people calling me up, including the pharma, where's the tissue? Where's the data? Where are you doing the biomarkers? So I think people are fully bought in. So when you get that collaboration and that overall engagement, I think it works well.
Yeah, if you have a good drug and a good trial, it's not hard to accrue to. I mean, that's been my experience. Do you have any thoughts on?
Well, yeah, good drugs make everyone look smart. I'd like to go back to something that Theresa was hinting at earlier on, which is that we need to think of the clinic as the final laboratory. Many of my laboratory colleagues will use the phrase scientists and clinicians to talk about two inherently different populations. As someone who's had both a laboratory but also done a lot in the clinic, that's always appalled me. And it's appalled me because the clinic should be the final laboratory. The problem is we regularly don't allow it to be the final laboratory. We do tiny studies. Those studies either have no biomarkers or poor biomarkers. They're frequently not done across institutions. And frankly, it's really hard to get pharma to support the laboratory parts of it in most cases. And because of that, we still have this divide between scientists and clinicians. I think if we allow clinicians to be scientists, we're going to get there a lot quicker.
Yeah, the clinical experiment. Yeah. And I mean, I think you see that even in large pharma where there's a huge preclinical science, and then it's kind of, okay, now you guys get it to the FDA. And I think the more we can narrow that divide, the better off we'll all be. I don't know if we have a microphone or if, I guess we can't see you up here because of the lights. So if you could, raise your hand if you have a question. Again, I can't, or scream out or something. Okay. Well, then I have one more question, unless someone else has some burning questions.
I think there's.
Oh.
Hi, thanks. I'm actually really interested in the last comment you just made, Dr. Sledge. Like, so what are the challenges in actually enabling clinicians to be closer to scientists? I mean, there's a lot of data out there, and in the clinic, you see patients, you form hypotheses, and we seem to be actually moving more in the direction of that not being the case in some areas, whereas in other areas it's perhaps changing in the opposite direction.
That's a great question. The barriers are present in pretty much all academic institutions and pretty much the same across all academic institutions. And they come from the different demands that are placed upon those who do science in the clinic and those who do science in the laboratory. Laboratory researchers, by and large, are tied to grant cycles. Those grant cycles have fairly specific endpoints, even if you don't actually end up following those endpoints, you're paid to do something. And you're paid on an every 3, 4, or 5 year scale. You're required to submit a whole bunch of grants because you know that only a few are going to get funded. So your life is tied to that. So when a clinician comes to you and says, I have a great idea. I want to take over your lab for a year so that you will do assays on my clinical study. The average laboratory researcher appropriately says, yeah, right. You're going to pay for that? And of course they're not, because they don't have the money for it as well. The clinician, on the other hand, is tied to a different schedule, and that's the schedule of the clinical trial. From the time I think about opening a clinical trial, let's say even the simplest phase 2 clinical trial, to the time that trial finishes accrual, is frequently, I don't know, Kim, 3 years if you're lucky.
If you're lucky.
And you only have so many shots on goal if you're an assistant professor and are trying to get promoted. So these kind of divergent goals actually end up killing a lot of what should be good collaborations. How do you get over that? Well, smart institutions, of course, have cancer centers that support translational research and support collaboration between the laboratory and the clinic. Smart pharmaceutical companies support those collaborations as well. And I think that's what we need, is just simply more support. Partly institutional support, partly the support in the form of time so that you can actually sit down and talk to your colleagues.
Yeah, and I'll chime in. I think also there's this huge demand on the clinical scientist, having done it for 20 some odd years, which is your first job when you get up every morning is to take care of the patients that are facing cancer, and that takes a lot of time. And then on top of it all, to ask a patient to be part of your experiment takes a lot of time. And there's increasing demands, and we've got to figure out a better way to make it so that the patient can be a partner in the clinical experiment, but that the physician also has the time. I can tell you that poor accrual, even with the best drugs, sometimes results because you just don't have the time to sit down and answer the patient. I mean, we ask a lot of patients, right? And I mean, you think about, here's an approved therapy and here's a wackadoodle therapy, and you have a 50-50 chance of getting on the wackadoodle combination versus the approved. To try to talk a patient through that so that they can really make an informed choice takes a lot of time. And the reality is there's a lot of demands on the clinical scientist right now. So
Hospitals today are largely run by process engineers who are interested in the question of efficiency. How many patients can I get through a clinic in a limited amount of time so that I can maximize patient care, but also maximize the revenue coming into the hospital? Entirely understandable, by the way. That's not a criticism, it's just a reality. But the end result is that if you're a physician and you are part of that process, you actually pay a price, and frequently a personal financial price, for putting patients on study. At Stanford, I earn the same number of work RVUs whether or not I put a patient on study or not. So if it takes me 3 times as long to put a patient on study as it does for me to see the patient and give them some standard third-line therapy that doesn't work very well. It's actually against my financial benefit for me to put patients on the study. That's a very simple, practical barrier that afflicts clinicians, clinical researchers across the country.
Yeah, I knew you would have something to say about it, so that's why I brought it up. And I think, the reality is that problem's even more amplified for community-based practitioners, where truly salary is somewhat dependent on the number of patients that you see. And that's why fundraising, whether it be academic pharma collaborations or foundation collaborations, I actually think is one of the solutions for to answer your question, which is to empower the clinical experiment to be even more fruitful and productive. So I think I'm going to call it. I want to thank, is there one more question? Sure.
I had a question on biomarkers. Do you think blood-based biomarkers are getting to the point where are they ever going to get to the point, do you think, where tissue, tissue's harder, it's hard to get, biopsies, add an additional element for inclusion criteria, is blood-based getting sensitive enough to have it be a pre and post where it can predict response? Do you think we'll get there?
Well, I want to say one thing about the tissue, because it actually, as much as we talk about tissue being hard to get, my experience has been, especially with the current minimal tissue requirements, it's not so hard to get. And patients, when you stop, this is my Kim Blackwell, not Lilly perspective, when you stop and think about what we ask patients to do, we put portacasts in them, we make their hair fall out, we make them throw up, we make them constipated, we make them come to the clinic once a week. Asking a patient to donate tissue so we could understand better what's driving their tumor, it has not, in my experience, been the rate limiting step. And I think we're starting to feel more comfortable with that, which is that compared to all the other things we ask patients to do, if they understand the risk, and it depends on where the cost comes from, they're more than willing to provide tissue. And usually patients that have tissue to biopsy are the ones that actually need the decisions made based on tissue. So that's kind of what I meant by the tools in the toolkit. I actually think the blood-based biomarkers are very close, if not already here in some components. I think what we're doing with some of the exosomes and cell-free DNA, some of which we've heard about today, are very close. That doesn't mean that we can exclude tissue. We're going to have to continue to get it to validate some of these tissue-free biomarkers, but that's exactly the tools that I think we've added to the toolkit that we didn't have even 5 years ago. So I don't know if George or Theresa, you want to comment on that?
So we're already there for kinase-based therapies. If you look at osimertinib in non-small cell lung cancer for EGFR mutation, there's already an FDA-approved biomarker for that. Alpelisib for PIK3CA mutations. There was a wonderful presentation, a couple of presentations earlier this year, saying that the blood-based biomarker actually outperforms the tissue biomarker in terms of predicting benefit. So I think we're already there for kinase-based therapies. I'm not at all sure that we're there for IO-based therapies yet, in large part because I'm not sure we have very many good biomarkers for IO-based therapies.
I mean, I've always been a fan of minimally invasive because while the tissue is very informative and we can now get biopsies, it's still a very small snapshot, and given the tumor heterogeneity, a single poke isn't necessarily representative of the disease. I think there's a lot of exciting data coming out in blood, particularly with the circulating tumor DNA and different machine-based learning. There's some nice things coming out. And then imaging, we heard a talk on that as well. We can't forget that there are other ways to see the tumor. So these PET-based imaging tracers, we saw one talk today, and there's, I think, one talk on Wednesday from ImagineAb on their CD8, which might give us a better look at T cells in the tumor.
Yeah, I echo your enthusiasm about imaging. I think it's going to be where we need to go. It's just going to be challenging because it requires multimodality of researchers to pull it all together. So, all right, I want to thank George, Theresa for joining me. Thank you all for staying for the rest of the day. And we've officially, I wish I could ring a bell or something, but we've officially made it through day one of the Precision Medicine World Conference. So thank you.